Good afternoon, everyone. Thanks for joining us today. My name is Emily Silva. I'm the NECAN coordinator. Welcome to the 2023 NECAN webinar series, in which the NECAN Steering Committee is gathering information on monitoring priorities for the Northeast. This is our fourth webinar in this series, and today is part 2 of OA and climate. With the assistance of this series and our presenters, the NECAN Steering Committee will be working on the development of regional monitoring plan for OA, and these webinars will serve as an informational resource for them as this plan begins to come together. Updates on this webinar are shared through our mailing list and up on our website. At the conclusion of the presentations today, the steering committee will be asking our panel some questions, and then we'll open it up for a more general Q&A following that. During that Q&A session, please feel free to use the raise hand function on the bottom of your screen, or you can submit questions and comments in the chat. First up, we'll hear from Rob Holmburg, a research scientist in marine carbonate chemistry and a designer of the OA lab at Downeast Institute. Rob is leading a series of groundbreaking experiments involving various Gulf of Maine bivalves and crustaceans, with a focus on present day and forecasted climate conditions peculiar to Eastern Maine. To that end, his work entails developing research partnerships with local shellfish farmers and authorities, exploring mitigation options in both ocean and hatchery, bringing the most up-to-date seawater carbonate chemistry analysis techniques to the region, and applying those techniques to monitor conditions in the marine environment where limited data resources exist. So you should be able to share your screen now, Rob.
Okay, great. All right, can everyone see my presentation?
Yep.
Great. All right, so today I'd like to present to you my presentation entitled Monitoring and Mitigating Sediment Pore Water Acidification on Main Tidal Mudflats. So my domain when it comes to OA monitoring is the main tidal mudflat, and it's a system that I feel doesn't get a lot of love when it comes to OA research relative to the water column. But it's a very interesting, very productive ecosystem. It also supports some very important fisheries, including the softshell clam, which is worth north of $16 million annually, as well as the quahog, which is worth $3.5 million, and other things like the razor clam, which are smaller fisheries. And I can't really talk about conditions on the mudflat without talking about the softshell clam decline. So softshell clam landings have declined by 75% over 40 years. Just a really precipitous decline. And this figure shows you the landings in millions of pounds between 1940 and 2020. And the drop in softshell clam landings coincides with a rise in coastal ocean temperature. And the biggest reason for the decline is predation by the invasive European green crab. So this is something the DEI has done a lot of research on. When we put out clam recruitment boxes, the number of clam recruits we find in the boxes at the end of the growing season declines with the mass of green crabs in the boxes. And we've also run experiments where we've covered recruitment boxes with different kinds of netting to exclude crabs. But what we found is that it's not that the clams are failing to settle and recruit, it's that they're settling and then they're getting demolished as soon as they settle. But of course, in the coming years, we're expecting ocean acidification to be another stressor on the clam fishery. And so on the left are the carbonate
Oh, Rob, you froze. You might have to take your video off. Uh-oh. Did we lose him?
We might have. If it persists, we can move on to Brendan and then maybe come back to Rob if he's able to find a more stable connection. Brendan, would you be able to jump in?
Yeah, I should be able to.
Okay. Hold on just one sec.
Should I go ahead and try to share screen?
Yeah, I think it looks like he logged out. We could give him a minute and see if he calls back in, or we could just move on to Brendan. Sam, what do you think?
Just
Yeah.
1 minute, and then we can move on to Brendan.
Those owls that you have in that room, Dwight, are really cool, but I'm wondering if we move too much, is it going to pick up the movement on the monitor and start thinking that I'm talking?
There is an ignore thing that I usually put up there, and it ideally ignores that part of the room.
That's
But
cool tech.
kind of. It's good when there's a group of people in here, but when it's just me, it's more complicated than it needs to be.
That's fair.
The only way to control that is through the app on my phone, and nobody else in my office appears to have the app, so I'm the only one that has any control over it.
All right, Brendan, it looks like it's you.
Yeah,
Try to
hopefully,
share.
hopefully Rob can reconnect after. Oh, I should introduce you. Hold on just one sec. So first up, we're going to hear from Brendan Carter. Brendan studied chemistry at UC Berkeley before attending graduate school in marine chemistry in the Dixon Lab at the Scripps Institution of Oceanography. After graduating, he did a postdoc with Jorge Sarmiento in Princeton and then took his current position as a now senior research scientist with the University of Washington's Cooperative Institute for Climate, Ocean, and Ecosystem Studies. He's the Cooperative Institute PI with PMEL's Carbon Group and leads and participates in several ship-based measurement programs, including the West Coast Ocean Acidification Cruises and the US Go Ship Cruises. He's heavily involved in ocean carbonate chemistry data synthesis. His scientific hobbies are quantifying anthropogenic carbon globally, estimating seawater properties using empirical and machine learning approaches, and asking questions related to marine carbon dioxide removal efforts. Take it away, Brendan.
Great. Thank you. And thanks for the introduction and the opportunity to kind of share some perspectives on these topics. I believe the prompt, I was sent a bunch of prompts. So I tried to kind of structure this talk around those prompts, and they were all focused on the anthropogenic carbon and marine carbon dioxide removal as kind of focus areas and the potential needs for monitoring these two sets of questions. I'm going to spend a few minutes, I'll start my timer here to make sure I don't get too off base. I'm going to spend a few minutes just kind of comparing and contrasting these because the perspectives I'm going to share initially really shape the answers to the questions that were posed for this talk. Anthropogenic carbon and marine carbon dioxide removal have a couple things in common that are very important. Clearly, they're both related to human impacts on the environment. Maybe less obviously, however, and very importantly from a perspective of monitoring, is that they're both very abstract quantities and they're actually unmeasurable, I would argue. When we're talking about what is anthropogenic carbon, it is the difference between our reality and another reality where humans had never emitted carbon. So it's kind of this one down here, or this is our planet here. And then with marine carbon dioxide removal, we're asking a very similar set of questions, which is what's the difference between our reality and a world where we had tried to implement some marine CDR effort. It's possible that in the future, we're going to be on this planet and kind of looking back at our original one and wondering how different those two are. But my point being here is that until we have a sensor that can measure both our reality and an alternate reality, ARPA-E, I'm looking at you, that would be high risk, high reward. Until we have that sensor, these are basically unmeasurable quantities. And so we need to find ways to estimate them rather than try to directly measure them. There's a difference between them, I would argue. For both of these, we really are considering all timescales, but for anthropogenic carbon, we're mostly asking questions about the present and the past. How different are things today from what they used to be or what they would be without this? And for MCDR, we're mostly today asking questions about the future. If we were to implement this, how would things be different? And that distinction actually shapes the answers to the questions as well in a pretty meaningful way. Okay, so NECAN presumably focuses on some time series. And then the question is obviously, well, okay, we didn't used to have as much anthropogenic carbon as we do today. So why can't we just look at time series and use that to estimate anthropogenic carbon impacts? And the answer is that we can, but the problem is that there's a villain in this tale. I don't know why I'm villainizing natural variability. It's very natural, but nevertheless, it is a problem here for that approach. You can think of natural variability as kind of the noise to the signal, which is the long-term trend that we're looking at. This is 3 examples of time series. We have the bats mooring up here, not a lot of PCO2 variability, a nice clean seasonal cycle. CCE2 mooring, you see some seasonality, but really you're mostly seeing subseasonal variability, tons of variability, about 100 microatmospheres swing up here, several hundred down here. And let's superimpose on that the kind of anthropogenic trend that we're looking
They're
at.
corded. They're white corded.
Oh, Katie, I got some mic noise from you. So when you superimpose on that the kind of anthropogenic trend, it's very small. Then we can
That one's
think.
been out for a while.
I'm going to pause.
She's been drinking it.
Thank you. Okay, then we can, this is the same sort of story over here on the right. By the way, can you see my cursor? Can someone nod? Okay, thank you. This is the same sort of story over here, but now with ship-based hydrographic data. We're only getting out there once a decade, but this is kind of going from Antarctica over to Alaska, the upper ocean. If you were to just directly plot the change in the dissolved inorganic carbon, this is what you'd see. It'd be a whole lot of red and some blue and big swings, very difficult to interpret. And that's because, once again, the anthropogenic signal, which is just this little red arrow, is getting lost amid natural variability. Same story with marine carbon dioxide removal efforts. This is now output from the simulation where we added a ton of alkalinity to Unimak Pass, Alaska, and kind of looked at what we expected the changes in the seawater properties up there to be. And over on the left is the signal. This is the change in realities. It's about 3/5 of a micromole per kilogram of total alkalinity, and about 1/2 micromole per kilogram of total DIC at some distance away from the release. Then when you compare that to the variability, it's 200 times that, no, 300 times that in DIC and many times that for alkalinity. So once again, the problem is that when we're thinking about the science that we need to do, it's really a very difficult problem to measure. We can't measure it directly. And when we think about using time series, the signals are going to get lost amid natural variability for a lot of applications. So these are the real challenges. Luckily, there's if there's an antagonist, there must be a protagonist, and we have plucky observationalists and plucky modelers on our side. So we can do things like deseasonalizing the data. I'm not going to explain all these panels, but the point here is that when you try to remove regular signals, if you have frequent enough measurements, you can try to characterize those regular signals and then try to remove them. When you do that, you can start to get at detecting these long-term trends. Similarly, for cruise work, we don't have frequent measurements. We only have measurements once every decade, but we can leverage the fact that we are also making co-located measurements of climate quality, other parameters. For instance, we can use changes in oxygen to infer how shifts in biogeochemistry might have changed the DIC, shifts in temperature and salinity in order to infer how much heaving and shoaling of water masses might have displaced DIC. And when you do that analysis, either kind of based on first principles or the approaches that I take, which are typically empirical, you can actually pull a signal out that is the anthropogenic transit or trend, but only under certain circumstances. And I'll talk about that further on in this talk. There's also ways to estimate anthropogenic carbon based on looking at tracers that don't have any natural variability. I'm thinking CFC11, CFC12, and SF6. Those do not naturally exist. So when you find them in the ocean, the natural variability is very small. Then you can use those, fit them, infer atmospheric timescales of contact between the interior ocean and the atmosphere, convolve that with a changing atmospheric history of CO2, and use that to get a pretty good estimate of anthropogenic carbon. There's a lot of folks who work on that. I'm starting to get into that space increasingly. So for marine CDR, this is where that difference starts to matter, because we're mostly interested in the future impacts. Not only can we not measure the impacts of marine CDR directly, but we can't even get to a time in which we could measure those impacts directly because we're worried about what's going to happen in the future. So this is really the domain of modelers. This is kind of what models are great at doing. When we start thinking about monitoring MCDR impacts as they are implemented, I would also argue that modelers need to play a big role there, because they're going to tell us how we can design our monitoring systems and what we can possibly hope to measure. We need model simulations of the global impacts of these kinds of MCDR implementations. The regional impacts will help us, decide what monitoring assets we need, and then the local scale impacts as well. I think each of these three sets of models are going to be answering very different and very critical questions. How much impact do we expect to have, and on what time scale? What are kind of the regional implications, and can we potentially use this to regionally mitigate ocean acidification? Local scale impacts, if we're dumping huge amounts of alkalinity right in this target area, or alternatively, if we're moving huge amount of CO2, dumping a huge amount of kelp, all these different techniques that are being proposed. What are the local scale impacts? Are we going to be maybe mitigating ocean acidification locally? Or are we going to be creating a Superfund site? We need to know these before we start actually implementing them. And both of these scales of modeling will help us decide how we can possibly hope to monitor the implementation and what we need to be targeting. Okay, so I'm now about 9-10 minutes in, and I'm just starting to get to the prompts. And I'm going to further complicate it by trying to separately answer the question for both coastal and global environments for some of these. Okay, first prompt: what is the most important decision, use, or application for monitoring within your theme? For global anthropogenic carbon, it's just where is the carbon going? Right now, there's about a 0.7 petagram per carbon per year, petagrams of carbon per year mismatch between kind of global model-based estimates, as represented by this purple line, and global observation product estimates, as represented by this green line, in terms of estimates of the global flux of CO2 into or out of the ocean. That may not seem like a ton, but that means that if our goal is to track where carbon is coming from and going globally in the hopes of using that information to help us meet our climate accords, we are off by about one set of European emissions. So we're a long ways from being able to track down individual kind of countries' emissions and whatnot. We are missing a continent at this point. For coastal anthropogenic carbon, I think the questions are a little bit different. It's less about kind of bean counting for CO2, and it's much more worried about what impact has the anthropogenic carbon accumulation had on that coastal chemistry and the ecosystems that reside there. For NECAN, you can maybe take a slightly more nuanced approach, which is, has that anthropogenic carbon pushed ecosystems beyond their envelope of natural variability? Are the organisms acclimatized to the conditions that they're experiencing? And there's lots of questions along those lines. So I think that begins with, you just need a good way to estimate the anthropogenic carbon and then propagate that through the carbonate chemistry to infer how different things are from what they used to be. Going back to marine carbon dioxide removal, the most important question, I think, is, are the models adequately capturing the processes that we need them to be? If the answer is no, then how can we refine those parameterizations? If the answer is we don't know, then we need to go to our third backstop, which is we need to think through, there are many processes that are captured in models that may be relevant to marine CDR. In some cases, I think we're very worried about uncertainties when we don't necessarily need to be, because we can code up the huge range of uncertainties in the processes that we're looking at and then see, okay, what impact does this actually have on the questions that we're asking? How much less effective is marine CDR for, when you have excess calcium carbonate precipitation, for example? And I think with those model simulations, we can start to triage the things that we're worried about. If we can put in those huge uncertainties and show that it doesn't, rise to the level of first order uncertainties, keeping in mind that people who do carbon accounting have very high appetite for uncertainty. There are people planting trees not knowing whether or not there's going to be a forest fire that burns them all down the next year. This is a way of saying, as long as we kind of understand the bounds of the uncertainty, we might be able to stop worrying about some of these. Okay, for coastal marine carbon dioxide removal, I think a lot of the questions are going to be focused on, again, is the model good enough to assess marine CDR? But then also, are the local impacts something that we're going to be able to detect? This is more output from that alkalinity edition, Unimac Pass, Alaska. The point here is these are different pH changes and kind of contours on that. I'm going to further simplify this to kind of break it down into a blue area, a red area, and a yellow area. Blue represents 0.0002 pH changes. So that's not measurable. That's about half of our best possible precision, never mind accuracy in pH measurements. Then we've got the red area, which had changes 0.002. That's measurable by some techniques, not all, but it's definitely not detectable. Natural variability will swamp that signal, and we'll have no way of knowing whether or not the change that we saw was due to what we did or just natural. Finally, we've got this tiny little yellow dot. And this is the area over which the changes are 0.02. And that is measurable, definitely, and detectable, possibly, depending on how much natural variability there is. So my point here is that we really need to have a good sense of where we're putting our observing assets and what our plan is for separating out natural variability from the signal that we're looking at. What needs to be monitored to support this? This is where I start getting a little bit more hand wavy. For all anthropogenic carbon estimates, I'm going to say we kind of need more measurements along the lines of what we've been providing in the past. For the repeated transects, looking at accumulation over time, temperature, salinity, DIC, oxygen, alkalinity is a maybe, micronutrients, salinity or silicate, phosphate, nitrate, all of those co-located measurements are needed to figure out what fraction of the DIC change that we measured is due to natural variability. I'm also going to say at least one set of these kind of transient tracer observations is really helpful. That gives us a baseline that we can use to kind of quantify changes over time. And then I wanted to throw this on here just as a prompt for discussion if people are interested. I'm not adding carbon isotopes on here. And the reason why is I think that they're actually answering a different question. I view anthropogenic carbon as how much extra carbon is there because humans did what we did, whereas carbon isotopes are actually telling us how much of the carbon that is there came from humans. And these are abstractly different, but importantly different questions, and they actually arrive at pretty different answers. What do we need for marine CDR? I would argue we need mean and variability understanding of just about everything. And why am I saying everything? It's because there are many different approaches that people are currently advocating and possibly even starting to try out for marine CDR. And they all tie into different processes in the ocean, different biogeochemical processes, air-sea gas exchange processes, physical mixing processes. So to really prepare for that, we kind of need to understand as much as we can. And this is just me doing a brain dump of things that I can think of what we might need to better understand before people are ready to start doing MCDR in an area. I said more narrowly, but really I meant more broadly. What we need is whatever's going to help us refine the models in order to prepare for a future in which people are relying on these models to quantify the impact, likely impact of a process, and also quantify what they need to be monitoring. Where is the most important place to make these measurements for anthropogenic carbon? I would say please continue time series that you have. Time series are really very useful for kind of figuring out these long-term trends. One exception for that time series is that if you don't have SF6, CFC11, or CFC12, it would be great to have a baseline for that. That said, I think NECAN is focused on an area that does have a couple sets of those measurements. But if you could add them to East Coast OA, that kind of thing, I think could be useful. More broadly, again, wherever you need anthropogenic carbons, you should start estimates. You should start making measurements there. Marine CDR, again, the correct answer is the second one. Ask the modelers what they need. That said, there is one set of people who are advocating an approach that doesn't involve models for monitoring MCDR. And they're saying, okay, get a lot of measurements, the background state and variability, construct an algorithm that kind of allows you to predict estimates from, or predict properties from one another. So this is kind of an example. This is a global open ocean alkalinity regression, where this is the estimated alkalinity over here, and this is the measured alkalinity, kind of a comparison globally. The thinking there is that you might be able to say, okay, we know we have a pretty good estimate of what the alkalinity would be in the absence of this process. So let's apply that estimate and let's make a measurement. And then we can say, hey, the difference between our estimate and measurement is probably due to this MCDR application that we put in. That said, the problem is that except for really huge signals, just due to ocean mixing, that change is pretty rapidly going to end up back in that envelope of uncertainty in the estimate. So it's definitely not a silver bullet. I think models are still going to have to be involved. But this might be one way to kind of still monitor local scale impacts. How often do we need measurements? I've got like 1 minute left. Is that right? How often? Anthropogenic carbon, I think about this in a single signal to noise context. The noise in this case comes from uncertainties in the measurements and methodological uncertainties. When you combine those, you get a signal that corresponds to a noise. Sorry, when you combine those, you get a noise that corresponds to a signal, which is about equivalent to the anthropogenic carbon accumulation after 5 years in the upper water column. So you don't necessarily need more frequent measurements than every 5 to 10 years. That said, the signal, the noise goes down as per the square root of the end realizations of that cruise that you have. So it does help to have more frequent measurements. And there are some places, especially off of Japan, where they're doing cruises every couple of years and they're getting really fascinating records of natural variability in anthropogenic carbon from it. For anthropogenic carbon, I would say that climate quality data is required. And that comes back to that last argument. The noise is really limiting what we can do. And the noise comes from uncertainty in the observations. This is too much to talk about, but it's really just saying that right now we've got an effort trying to estimate how much uncertainty there is in the GLODAP v2 data product. And what we're finding is that taken collectively, we're not quite hitting climate quality with GLODAP v2. More narrowly, when you focus on subsets of cruises, one of the best subsets that is meeting climate quality, it includes the US OA coastal cruises. So that's good news. Monitoring needs for MCDR, again, ask the model.
Hey, Brendan, was that the coastal OA cruises, does that include ECOA? Is ECOA hitting climate quality?
I'm not sure. I'm not sure if they're in GLODAP, and also I can only make these statistical generalizations for huge collections of cruises. So even if it did include them, I can't promise that any one cruise is not or is meeting GLODAP or climate quality observations. Does that make sense? So yeah, I wish I had a better answer for you on that. Okay, for MCDR, I'm going to say ask the modelers. That's the summary of the slide. Can this augment existing monitoring? I think I'm over time. So actually, I'm probably going to skip ahead and just say the final part. I've got some other slides we can talk about later if people are interested. But I just wanted to thank everyone for your attention and make sure I don't steal too much time from other folks. So thank you.
Thanks, Brendan. Sam, did you have a question, or is that applause for his presentation?
There's applause.
Oh, I had a feeling.
Yeah, we were
thanks,
waiting
Brendan.
on questions, right? Emily, yeah.
Yeah. So, yeah, Rob, I'll let you go ahead and share your screen.
All right, thank you. All right, so I'll pick up where I left off. My apologies for the hiccup. I didn't actually lose connection. I was resting my phone on the dashboard and it overheated in the sun and my phone shut down and it was providing the hotspot that's powering my connection here. But I have the AC on now. All right, so I left off talking about the normal conditions on made mudflats in the pore water. So relative to the water column, sediment pore water is very acidified and very undersaturated with respect to aragonite. The reason, of course, being that the mud is full of organic matter, which microbes are remineralizing and adding that CO2 back into the system. And so there's concern that ocean acidification will not only acidify the water column, but also reduce sediment pore water pH and omega below their natural minima. So in other words, the infaunal organisms that are inhabiting the mud are adapted to some level of acidified conditions, but there's a fear that OA will push it further. And also, as far as I know, I'm the only one that's monitoring this regularly in Maine. All right, so there's some evidence that pore water acidification will impact bivalve condition and behavior. A lot of which comes from Mark Green and Jeff Clements. So in 2009, Mark Green found that juvenile quahogs exhibit dissolution mortality in undersaturated sediments. Their shells are literally dissolving and they're dying as a result. And it also impacts infaunal bivalve behavior. So So for example, Green et al. 2013, juvenile quahogs actually burrowed more and settled more in sediment conditions that were more saturated with respect to aragonite. And similarly, soft shell clams exhibit a similar pattern of increased burrowing with higher pH. All right, so most of the measurements of pore water carbonate chemistry up till now come from a handful of measurements from a single point in time. So for example, Green reported pH between 7.03 and 7.05 on a mudflat in West Bath, Maine in 2007, with a corresponding omega of 0.25 to 0.26. And in the same paper, Green identified a critical threshold, an omega threshold, below which quahogs begin to dissolve and die. And that threshold is under 0.6. And he also identified lesser softshell clam recruitment at lower omega. And some additional measurements from a different mudflat in South Portland. Green reported 40% of omega measurements below 0.6, and fewer hard shell clams and soft shell clams burrowing and recruiting at lower omega conditions. So going back to the question of what should we be measuring? Well, we're interested in the carbonate chemistry of the pore water, and the most accessible measurement is pH. But typically you also want to get that second parameter so that you can calculate the whole system. And so typically the way researchers have done this in the past is they'll go out in the mud and they'll take a handheld pH meter with a probe and effectively submerge the probe in the pore water and record the pH. This can come with some challenges. For example, pH probes are really intended to measure the pH of the fluid medium. So when you're sticking them into the sediment, you might get a lot of noise. And in my experience, it really takes some time to allow the measurement to settle before I feel confident that I can record it as an accurate pH value. And then other carbonate chemistry parameters typically come from scraping samples of mud and storing them and centrifuging them to separate the pore water from the sediment, and then analyzing the pore water for something like alkalinity, DIC, or what have you. And so I mentioned the importance of measuring two parameters. The bulk of the existing research only reports pore water pH, but pH alone doesn't really give you the whole picture. Of course, the other parameter that's really important, known to be really important for bivalve condition, is the saturation state of aragonite, or omega. But you can combine any two of these parameters to calculate the remaining parameters. But I generally think of pH and omega as being the most important parameters, and so that's what we're after here. And when I started measuring this stuff in the sediment, I really wanted to do it above board and get high quality, accurate and precise measurements. And so when you're measuring pH potentiometrically with a probe, if you're measuring seawater or pore water, it's really important that you're calibrating that probe to artificial seawater buffers. TRIS is most commonly used, but if you want to do a 2-point calibration, you can do 2-aminopyridine as your low buffer and TRIS as your high. And this is really important because the salinity of the seawater and the pore water interferes with the action of the pH probe. And so if you were to compare measurements from a probe calibrated to standard 7 and 10 NBS buffers calibrated at 25 degrees Celsius, and compare those to another probe that has been calibrated to synthetic seawater buffers, you're going to see an offset which will average around 0.15 units higher than the real pH, but it can, in my experience, it's frequently higher than 0.3 pH units. And so from my perspective, if we're collecting all this data and we want to be comparing it to measurements that other researchers have collected in different parts of the state, then it's really important to get measurements that are both accurate and precise so that you can enable a fair comparison. And then, of course, if you're measuring another parameter like alkalinity or DIC, it's really important to compare the accuracy of your measurement to a reference material. I think we're all familiar with Andrew Dixon's CRMs. That's what I'm using to calibrate my alkalinity measurements in the lab. All right, and so one of the challenges of measuring this stuff in pore water is that you can take a 50 mil Falcon tube and fill it right to the top with sediment and then spin it down, and you may only get 5 mils of pore water out of it because the samples that you're taking are mostly sediment. And when you're working with that little sample, it's really challenging to measure things like total alkalinity or DIC. And so I looked through the literature, and one technique that researchers have used in the past is running your typical open cell total alkalinity titration, but replacing the pH probe with a pH microprobe. I purchased one a couple years ago to try it out, and I broke it within 3 days. If you think standard pH probes are fragile, microprobes are just this really thin strand of glass, and the moment they touch anything, they shatter. So instead, I've been leaning on spectrophotometric methods for this stuff because spectrophotometric methods are somewhat scalable. And so if you adapt a spectrophotometric total alkalinity method from the literature, the paper might call for 100 mils of sample or 300 mils of sample, but it's possible to shrink that down, and we're doing it with as little as 5 mils of sample. which makes it effective for pore water and gives really accurate measurements. But of course, doing that requires some DIY, making your own instrument inserts and coding your own macros and whatnot. So I mentioned that as far as I know, I'm the only one who's monitoring the carbonate chemistry conditions on main tidal mud flats on a regular basis. My two study sites are both within Beals, which is where DEI is located. One of them is on Beals Island and the other is on Great Wass Island. And I kind of chose them because the Perio Point location is sort of a little more open to the ocean. It's open to Moosabec Reach, which is the body of water between Beals and Jonesport on the mainland. Whereas the other location, Dobbins Island, is a little more enclosed in a cove, and it traps a lot more seaweed, which becomes organic matter. And so the conditions are different, which makes it interesting for comparison. And here's what I got from the year 2022 season. One of the prompts for this seminar was, is there any particular season that's really important for monitoring this stuff? For us, we're really interested in the biological endpoint of clam abundance on the flats and clam condition. And so I've been measuring this stuff throughout the clam growing season, which is roughly between April and November. The larval clams are settling in the spring, and then they're growing through the fall. And so if you look at these figures, on the left we have pore water pH and on the right we have pore water omega. Both locations are displayed in each figure. The white boxes are the Dobbins Island site and the gray boxes are the Perio Point site. And so we're seeing some marked differences in pH and omega, not only between both sites, but also from month to month. And I mentioned before that a lot of the existing measurements of pore water carbonate chemistry are from individual points in time. But with my project, I felt it was really important to monitor it over a longer time period because it's liable to change from month to month throughout the season with the cycle of primary productivity and microbial decomposition, just like you see in the water column. All right, another question from the prompt was, where should we focus our efforts? So if I'm being honest, I chose my study sites because they're conveniently located down the road from DEI, and I'm able to get there for the early morning low tide and then sample, and then bring the samples back and preserve and analyze them. But also being in the Downeast region, you know, we're a little further north and the conditions are a little bit more acidic and undersaturated. And so according to Xtrom et al. 2015, the Downeast region of Maine, including Hancock and Washington counties, are going to hit those critical omega thresholds earlier than the rest of the state. And also we have a higher social vulnerability because so much of the economy in our counties is dependent on the fisheries, including clam landings. And so one approach for choosing where we should focus our effort is focusing on the most vulnerable communities. But having said that, I'd like to expand to different parts of the Maine coast. My monitoring project is kind of piggybacking on DEI's clam recruitment monitoring network project. And so they're putting out all these clam recruitment boxes all up and down the coast between Wells and Sabayak, which is even further north on the coast than Eastport, if you can believe that. But those boxes make for really nice replicates, so I can go out month after month and measure these conditions from the same places. And so I have part of the Downeast region covered, but I'd also really be interested in expanding this monitoring effort to mid-coast Maine as well as southern Maine, so that we're able to compare the conditions and see if they really are different from region to region. Another thing worth considering when measuring carbonate chemistry parameters in seawater or in pore water is the matter of uncertainty propagation. So you can take any two parameters to calculate the whole system, but not all pairings are created equally. And so I've been measuring pH and alkalinity and using those to calculate omega. But according to this paper by Oradol from 2018, that's actually the third best pairing of carbonate chemistry parameters out of 6 because of uncertainty propagation. So in other words, you're measuring 2 parameters and that's your point A, and then you're doing a lot of calculations to get the other parameters, which is point B. And the further, depending on how uncertain your initial measurements are, and depending on how far you have to go to get to point B, you're going to end up with differential accuracy in your calculated parameters. And so compared to the pH and alkalinity pairing, the ideal pairing is actually alkalinity and DIC, which has a lower total uncertainty when you're calculating omega. So one direction I'd like to move in the future, and that I think we should move in general, is ditch the pH probe entirely and just collect mud samples and then analyze the pore water from those samples for alkalinity and DIC. Of course, that'll necessitate scaling down a DIC method to work with such small sample volumes. And that's something I've been working on at DEI. And so finally, this isn't really the same as carbon removal. It's more of a Band-Aid that we might be able to apply to pore water acidification. But one technique that's been proposed as a method for buffering pore water acidification is spreading crushed up oyster shells or oyster shell hash. And so at DEI, we've done this using a machine called it's a glass bottle crusher, but it's effectively a miniature hammer mill. And you can put oyster shells in it, crush them up, and then if you sieve the shell crush, you can achieve different sizes of shell pieces. And so if you're thinking that sounds a lot like an interesting factorial experiment, well, that's exactly what we did on a mud flat in South Portland. Incidentally, the same mud flat that Mark Green collected some of his measurements from. And so we set out 120 different 1 square meter plots with different combinations of shell crushed sizes and shell densities, and also predator deterrent netting to keep out the green crabs. And again, we measured the pH and alkalinity twice in April, once in May, and then finally in October at the tail end of the clam growing season. And this is what we found. You can see a seasonal signal in both pH and omega. It's higher in May relative to April, and then it sinks lower than April and October, which again, I think ties back to the whole cycle of primary productivity. But we also analyzed it for the effects of the shell, and we found that any shell at all actually increased the pH in the pore water, but the smaller shell increased pH more. And then as for omega, we saw a similar effect of, well, not just any shell improved omega, but the smaller shell increased omega more. All right, so I'd like to thank you for listening, and thank all of our great collaborators at DEI and beyond, and I'm happy to take any questions.
Thanks, Rob. And thanks again, Brendan, as well. I'm going to hand things over to Chris Hunt to lead us through our Q&A session with our steering committee first, and then take some general questions following that.
All right. Thanks both to Brendan and Rob. Rob, especially for powering through your technical difficulties. I see Dwight has his hand up, but I'm going to take my prerogative and ask a question first, and then we'll come around to other folks. Rob, I want to start with you. I have great appreciation for the amount of work that goes into measuring the parameters that you're measuring in the environment that you're measuring them in. And so it's really labor intensive, both in the field and in the lab too. And so in thinking about if we were to expand pore water measurements as part of a network initiative, can you see any either technological advances or anything that would expand the capabilities of monitoring these things either in space or in time without having to like massively expand the number of person hours that are devoted to making these measurements, if that question makes sense?
Yeah, that's a great question, and it's something I've thought about a lot. It certainly is really labor intensive. Typically you have to aim for the morning low tide, which it might be 4 in the morning, it might be 5 in the morning.
Yeah.
You got to put your tall boots on and get out in the mud where it's difficult just to walk sometimes. And we've relied on my various interns as well as local shellfish committee members to help us with the physical labor. But I have thought about how we might be able to automate some of these measurements. And one thing I've read about that I wasn't really comfortable making a slide on yet, but it's possible to create a pore water well. And so you're essentially putting a pipe in the sediment, which is enclosed on both sides with mesh and might have a T in the middle.
Yep.
And so the pore water will actually diffuse out of the sediment and into the well, and then you're able to sample from the well. And I thought, well, wouldn't it be nice if we could just create a pore water well and then stick some kind of logger in there, like a hobo pH logger, for example.
Yep.
Or if you wanted to get more sophisticated, maybe we could engineer some kind of black box that'll do alkalinity titrations or something to get that second parameter. That said, even just having the well would make it easier to sample stuff. Even if you still have to go out, you could just draw water from the well and preserve it and probably get a much bigger sample for analysis.
I imagine you have to.
So,
I imagine you have to isolate it from the overlying water for when the tide comes in, right? But that's all technical stuff.
Right, for sure. So maybe you could have like a tall standpipe or something that exceeds the highest tidal height. So yeah, I thought about this. It's something I'd like to test, and one of the things that I would like to test is whether, we could dig the well and we could get the pore water that's collecting in the well, but are the conditions that you're collecting in your well the same as the conditions in the surrounding mud if you were to do this the traditional way?
Sure.
So that's a question that I have and that I'd like to test at some point in the future. But if we can get to that point, then maybe we could put these monitoring stations on various mudflats around the coast, and then we could really beef up our not our effort, but our data collection.
Awesome. Brendan, can I ask you a question?
Yeah, of course.
So, and I really appreciate your talk because it came at it from both ends, from the real broad scale and also the local scale. That was great. You, as sort of as an aside, you mentioned that you think that some folks are doing tracer analyses in our kind of neighborhood, either in the Northeast or on the East Coast. This is an analysis that I'm not super familiar with. Can you talk a little bit more about that, or about where we could look for information on that?
Yeah, so I could check the GLODAP data product to see kind of what's available in this region. Okay. I probably should have done that before the talk. I do know that GoShip runs several lines that come into and out of the US West Coast, and they always do.
Actually, we do. This is
Oh.
Kamika, it's Scott.
Ah, hi,
Perfect. Thank
Kamiko.
you. Yeah, maybe you should talk about that then. I think that would be really useful for me to hear as well.
I don't want to jump the line, but I have a related question. Is that okay? Do I just ask as the spinoff of that? Is that my question too about that was about the CFCs and SF6 or the carbon 14, in the DIC, those kinds of tracers. And so I don't know, maybe Kamiko can answer this one. I don't know, but like in these kind of seasonally ventilated kind of settings, like, is that something I haven't seen it kind of used in coastal waters much? Like, are there considerations there and like things that, or other tracers that might be more useful in that situation?
Yeah, that was a question I had too, because TTD usually used for the deeper waters, right? Like ghost ship type. And shelf coastal waters, we tend not to use CFC SF6. It's not that useful because, as you said, it's mixed so well. So that was a question I had. Yeah,
that's a really good point. So I'm coming at this from the West Coast, where when we have waters coming up, they're typically, from a ventilation perspective, somewhat similar to deeper waters. But yeah, certainly there's this awkward depth horizon with anthropogenic carbon estimation, where at the surface, this micro layer, you assume it's fully equilibrated. And then at around 200 meters, you start to have little enough seasonal variability that you can start to kind of use the traditional methods. And below that, you have the access to these ventilation timescale tracers, TTDs, as well as the kind of OSIM optimizations. But yeah, certainly in the uppermost water column, when you don't have upwelling, strong upwelling, it's challenging to estimate anthropogenic carbon. Fortunately, the closer you are to the surface, the less bad the assumption of complete equilibration of the atmosphere is. So.
Great, thank you. All right, I'm Dwight. We can turn to you. I know you've been waiting patiently.
Not about patiently, but I've been waiting. Yeah, no, that's great. Just to carry on with the CFC, I don't really have any more to add what I had in the bar. I think everybody's covered that. And now, after what Brendan just said, now I'm really befuddled as to where we go with that. Because do we have an appropriate tracer for this region that we can use to get at that? And maybe the carbon isotope approach might be the best we can do. I don't know, but like you said, it provides us a very different information, right? So let me come back to you on that. I do want to go back to Rob though, because I was real excited about what you presented, because I haven't given a lot of thought about a large systematic sediment geochemistry sampling program that has really direct application to an important marine resource. And I have a bit of a sediment biogeochemistry background myself, so I was like listening to the struggle you're having, going, oh yeah, I know those struggles. So first off, I used to work with very small volumes, including sediment. Small sediment volumes, and we used to approach it in the Morse lab using gravimetric dilution approaches, but it requires you to have very tailored standards worked up, so you're really narrowing that range and understanding that response. So I used a regular Ross pH electrode, but I just did a gravimetric dilution for alkalinity. And I even did DIC directly using a kilometer, using only 3 to 5 cc's. But again, I had a pretty aggressive calibration curve worked up for that. So it might be something to think about. But one big question I had, actually there's a few of them, is so how do you handle gas exchange? So it sounds like you're taking them up and you're centrifuging the sediment sample, but are you giving any consideration to gas exchange?
You mean gas exchange while they're in storage?
When you're spitting it through the centrifuge, right?
Yeah, it's something that I haven't really considered in depth. you know, they're filled right to the top. So it's my hope that there isn't.
Well, okay, so you got the whole thing filled to the top, I see, and so it's completely sealed, and so when you centrifuge it, you've just, so the water is just on top, so maybe you don't have any gas exchange in that case.
And I have to seal them extra well, because if I don't, then they'll leak all over the centrifuge.
Yeah, okay, yeah, all right, so that makes sense.
That's probably gas exchange, probably less of a worry for TA, but if you did want to move
Right.
branch out into DIC,
Move toward
then,
DIC. Yeah,
then it would be a
yeah,
consideration,
DIC
sure.
has some challenges. Yeah.
It would have similar effects, I think, on pH would also be sensitive to, you know, gas exchange as well. So that's just something to think about, but it sounds like maybe that approach you have might work. I do like, so we used to use lysimeters, which is what you're talking about, these pipes that would go in. And you can use lysimeters, and they're a bit of an effort to put in. They're usually like a silica gel around the whole outside, or there's different ways of doing it. So I kind of like that idea. But one thing I'm wondering too is, so what's the depth in which you're pulling the sediment sample from? Is it always consistent? Is it specific to the depth at which the clam resides? Have you looked at the sediment profile of the geochemistry in these systems?
Yeah, so the depth profile for a parameter like pH is pretty dramatic. And so I've been really careful to standardize the depth of all of my measurements and sample collections to within the upper half centimeter of sediment, because I'm really focused on the larval and early juvenile stages of the clams.
So
which,
then that brings
you know,
up
we have reason.
That brings up a further complication if you're looking at right at the very near sediment surface, and you're only going out at low tide, when the whole water mass is pulled off and popped the lid off of that cork, which is now probably doing a fair amount of gas exchange. So I would be very curious to know if there's a difference in that geochemistry for when the water is sitting over top of it versus when it's venting to the atmosphere. Right, so you might get very.
I had that same question about the inundation. If you had thought about that kind of period. Sorry, Brendan, I see your hand up. I just following on.
Yeah, it's definitely something I've thought about and that I'd like to test formally. What we've been doing is we're collecting the measurements at least at the same time relative to the tide, meaning like we go out in our boots and we wait for the water to recede, and then we do all the measurements and sample collection as soon as it recedes.
Right, so it's internally consistent, but I think if you're trying to emulate the environment in which they're recruiting in, you might get a very different environment when the water's sitting over the top of the system. And that's going to be, you know, and that's when they're going to be recruiting. Yeah, anyway, but I really like it. I want to talk more on it because I am curious about like the square acreage of how much area we're talking about in the region that's actually being clammed, or what it would take. So yeah, really interesting stuff. Sorry, Brendan, go ahead. You got a question?
Yeah, I'm not sure if I should be asking questions, but I'm kind of curious. So you mentioned kind of switching to a different carbon parameter. I'm wondering if it would make sense to do all three. And my thinking is in the context of organic alkalinity. Do you have a sense for kind of how much organic alkalinity is present in the pore waters?
Yeah, so this is something I've spoken with Chris a little bit about, and I have to assume it's a pretty high percentage, and I'm wondering how it could be impacting my alkalinity measurements. And so Chris is working on a project to characterize organic alkalinity, you know, which might help give me an idea of how much of an impact it's having, or how I might be able to measure it myself.
Rob, I wish I could tell you, but the great thing about organic alkalinity is there's no set method to measure it, and so there's no sort of accepted protocol and no accepted definition of it. So it's really the Wild West in organic alkalinity right now. But I could easily imagine that coastal sediment pore waters would have a bunch in it, right? And we have a couple different ways we can try to measure it, and we have some ways we're developing to try to characterize it. And so I think kind of like you're pushing the envelope on measurements in some of this coastal pore water areas, we're trying to push the envelope on how we can look at organic alkalinity. But yeah, Brendan, I mean, I love anytime anybody brings up organic alkalinity, because it is a little bit of the elephant in the room when you talk about total alkalinity measurements. So measuring 3 would be, I mean, obviously 3 is better, of course, right? But it would be a really interesting addition to Rob's project for sure.
Hey, Rob, one more thing. While we're talking organics, that, what'd you call it? It's the Clan Recruitment Monitoring Network. Some of the other data that they're collecting, are they collecting like organic carbon measurements from the sediment? Like, do you have some understanding of how much organic matter is in that? I'm guessing that whatever the pH is, or the saturation state at any given site, is probably dominated by how much labile organic carbon is in the system, right?
Sure.
So is that part of the data network's standard sampling? It's kind of knowing how much organic
So
carbon is in the
the CLAM Recruitment Monitoring Network project isn't monitoring any kind of chemical endpoints. They're only monitoring clam abundance and crab abundance in the boxes.
Okay.
So no, we don't have any organic measurements. The pH and the alkalinity measurements that I've been collecting are sort of the first step working toward that.
And so the sites, Rob, the sites you showed on your map were like where you would like to expand to monitor the chemistry of the sites that are right now just doing recruitment. Is that right?
Right. So all of those circles and stars on that map that I showed are mudflat locations where DEI has put down recruitment boxes.
Yep.
And how often are they getting out there? How often are they getting out there, Rob, to do those recruitment checks? The network? What's the frequency of observation?
They have a lot of related projects happening, but typically they'll put the boxes out in the spring and then come and collect them in the fall, and then they'll rinse all the sediment away from inside the boxes and sieve out any settled clams that are in there.
So each of those sites gets visited somewhere on the order of every 6 months. Is that right?
Yeah,
Roughly?
yeah.
Okay.
But the boxes stay put throughout the spring and summer and early fall. So I'm using them as my replicates when I sample.
All right. So I want to make sure that the other members of the committee have a chance to ask any more questions they have, but I also want to give everybody else in the audience an opportunity to ask questions as well. So before we open it up further, is there anybody else from the committee who wants to chime in? So going once, going twice? No? All right. I mean, I know that there was a nice point made by Nicole Price in the chat. Nicole, I don't know if you want to ask your question in person or just have me read it.
Can you hear me okay?
Yep.
Oh yeah. You could just read it. I'm just curious how we're gonna handle that kind of predictive uncertainty. I'm sort of desperate to know how we'll do it.
So this was kind of in connection to Brendan's point about carbon markets and about like reforestation efforts and stuff like that, right?
Yeah.
Okay, so Nicole said carbon, I'm just going to read it. Carbon markets are more concerned with risk assessment of the likelihood of a reversal, e.g. of forests burning down, or of upwelling or sediment disturbance. Uncertainty in deposition or storage rates are much more palatable. So Brendan, I don't know if you have thoughts about comparing uncertainties or comparing risks in terms of reversals versus actual removal rates.
Or if there's anything from the historical data that you've been looking through or the spatial data that lets you know how often a disturbance of the bottom or some sort of big upwelling event could happen.
What's the likelihood of stuff being brought back up to the surface over longer timescales?
Yeah.
This
And,
certainly
and
highlights.
projects are generally 10 years. Sorry, just to interrupt really quickly. It's not the 100-year timescale, it's a 10-year timescale.
Gotcha. So you're kind of asking in the context of the marine CDR efforts that are thinking of like sinking kelp or putting biomass on the ocean floor or burying carbon. Okay. For that, I certainly don't have an answer to the question asked of the likelihood of kind of those upwelling events in NECAN's area, but I do think that the modeling assets that are there probably could start to answer those kinds of questions. I do think that this does really illustrate the fact that MCDR brings together many different communities, and it brings together people who are used to thinking about money and carbon and offsets and risk, and it brings together folks like us who are used to thinking about circulation and biological cycling, and it really touches all elements of earth science because the strategies that are proposed, like I had to take a moment to ask us, are you talking about these kinds of MCR versus ocean alkalinity versus that artificial upwelling? So I just think that this is a caution for this community, which is that there are many different groups involved and we have to learn how to kind of speak one of those languages. Sorry, I don't have a better answer to the question you asked. While we're getting to the next question, would you mind if I shared my screen really quickly? I pulled up a map of CFC and SF6 observations.
Oh, please do.
So this is where we have CFC 11 observations. It's a little bit more dense than where we have SF6, which is the kind of tracer that's a little bit better for getting circulation nowadays, now that CFC 11 and 12 have been banned and stopped accumulating. So there are some observations in this area, but there are certain places that with, the most recent tracers could be hit again. And also this is about a year old, and I think there's been another cruise since then in this area. So,
but it looks like they tend to stop right at the shelf break or close to it.
That too. Seems that way.
Yeah, except for the Scotian shelf, it looks like Kumiko's going up on the shelf.
Yeah, we do. On the way back from Labrador Sea, we mainly use for Labrador Sea, but on the way back, we do section in Scotian Shelf when you measure them. And deeper part, like Emerald Basin, which is deeper than 250, then you can see the difference at the bottom, but it's very limited area. You can see actual difference.
But so I'm just wondering, can we do it through mass balance if we have an understanding of the primary sources of water mass entering the Gulf of Maine? I mean, we can get good CFC estimates in the Gulf Stream and in the Labrador Current. Maybe I'm wrong about that, probably not. I'm just trying to wonder, like, if we can't actually obtain it in the Gulf of Maine, can we at least quantify in all the water masses that fill the Gulf of Maine, and therefore work some mass balance to estimate it? I mean, we may never be able to do a direct measurement of it.
Total novice question. What's the depth of the Gulf of Maine, approximately?
I mean, the deepest part in Wilkins Basin is 250 meters or so.
Okay.
So,
does anyone here have a sense for how frequently it's ventilated? Because if this is ventilated annually or every couple of years, it probably this would not be the right tool for it.
It doesn't get mixed all the way down, I don't think, to 200 meters, but I think residence times calculations that I've seen have been on the order of years.
Yeah.
So it depends on what you're talking about, I guess. Like,
this might not be the right tool.
Yeah.
I might be coming here with my 4,000 meter global oceanography bias and thinking about the wrong approach here.
Blue water oceanography.
But we can do EMLR though.
Yeah. And there was a nice talk about that the last round around of these webinars for folks if they're interested in checking it out as well, an initial approach using that.
Yeah, Wiley Evans presented an approach, and so did Mingyu, right?
Xinyu, yeah.
Xinyu, yeah.
Right, but I think like Brenda was saying, there is a slightly subtle, well, maybe it's not slightly subtle, but there is a bit of a theoretical difference in what it is you're measuring, but that might be what we can get. But at the end of the day, I think what we'd want from an agency perspective is that if we see shifts in the ecosystem that we think are responsive to acidification. I just think of one day of the lawsuit that comes and says, can you actually quantify for me how much of this shift is attributed to human activity?
Mhm.
And that's what we'd want to be able to answer as an agency. Say, well, we see the shift, we see the ecosystems responding, and this is the percent of which humanity's activities have contributed to this change, right?
Yep. And, you know, I think we're furthest away from getting to what that effect is inside the pore waters.
Yeah.
But that's just my suspicion.
Yeah, no, I
Like,
agree.
you know, but getting there, we're getting there, building
Yeah.
pieces. Mhm.
When Brendan talked about, you know, deseasonalizing time series to look at long-term trends and the natural variability, when you look at the natural variability in Rob's pore waters, like in pH, you know, it's what orders of magnitude more than in overlying water. It's wild. So that's, yeah. So I guess I had a question kind of following up on Rob's stuff too. So Rob, you presented, and then this is coming back to your map again, because that's stuck in my head, but you presented sites you'd like to expand to in Maine, but so NECAN, you know, encompasses, you know, Maine is a very large part of NECAN, but not all of it.
The Northeast, yeah.
Right. So, you know, and you have a clam focus for now, but there, I imagine there are other shell fisheries around the Northeast that would also benefit from this kind of study. So do you have any thoughts about, you know, outside of Maine, you know, getting away from Maine a little bit, where else would make sense to try and establish monitoring if you had the resources that you needed?
Well, you know, anywhere there's clamming.
Mhm.
So that could include Rhode Island, where quahogs are a really big fishery.
Mhm.
I've been focused on Maine because of the, you know, the relative abundance of these large tidal mudflat systems that we have locally, and because they're such a big part of, historically a big part of Maine culture,
Sure.
as well as relatively large fisheries. Um, yeah, I have to say I haven't put a lot of thought into where I want to expand it outside of Maine.
Yeah, I mean, I also think about like other substrates besides mud, right? That there's certainly
Yeah,
pore water
sandy
chemistry.
soft bottoms, and yeah.
Right, right. So I should say, we intended to have another person present today, Chris Alger, who's from Dalhousie, who does pore water chemistry, but using cores, more offshore. So he's looking at different substrates like that, and he'll present in a later session. He just had a last minute fieldwork thing come up. So maybe he can inform that discussion a little bit, but I think that that's something that would be really interesting to think about. And
Yeah, that would be great.
I see in the chat that Katie O'Brien Clayton threw in, what about oysters too? So oyster fans always want to know how the chemistry is going to affect oysters too.
Yeah,
Obviously,
sure.
it's a different issue for wild oysters versus cultivated, right? Because cultivated oysters are usually raised. I mean, I've seen them raised in like floating boxes and in a hatchery and so forth. So the chemistry is much different.
Yeah, so around here, we do spawn oysters in the hatchery, and we'll deploy them in the ocean. And typically without cages, we'll just spread them on the soft bottom at a lease site. I just visited one this morning. There were just juvenile oysters scattered around the bottom. I will say that in the past, DEI has expanded our recruitment monitoring network north into Canada.
Okay.
Across the border. And of course, they have the same species we do in Downeast, Maine. Soft shell clams. That's another, I'm assuming Northeast also encompasses Atlantic Canada.
Absolutely.
Yeah, so that's another realm of exploration.
Yeah, you can go both ways. All right. There any
So,
more? Oh.
Can I just ask a quick question, Chris? So, I mean, I guess just tying, the large regional scale changes to the sediment interface. I'm wondering if there is any literature that maybe I'm not aware of, but it seems to me that where ocean acidification might more meaningfully impact the sediment geochemistry might be by means of changes in the biological carbon pump. And this is where getting back to the question I had about what do we know and understand about the labile organic carbon content of these sediments? Where is it sourced from? How much of it is dependent upon primary production? And if there are changes over time that we see in the Gulf of Maine driven by acidification, would that have knock-on effects to the amount of labile organic carbon delivered to the sediments of these clam beds? And that may have a more dramatic effect on their local carbonate chemistry than the direct effect of pH. And I don't know if we have an observing system that can interrogate that question, or if that's even if that's even a good question. I guess that wasn't a question, but a comment. So maybe Sam or Brendan can check. Is that completely off the mark? I mean, is there expectations? And maybe Rob knows this. Like, I don't know enough about where the organic carbon comes from that is present in those sediments, and what dependency it has on the geochemistry of the Gulf of Maine.
I think it's a great question, Dwight.
Yeah, I agree.
And do we have an observing system that is collecting the data that could interrogate that question?
If I could hop in, I actually have a comment.
Oh, yeah.
I would say that we're working on an observing system that might help, which is the genetic-based metagenomic and qPCR approach to query the sediments as to whether or not there are macrophytes that are contributing to the carbon content, but that could easily be adjusted to ask questions about any organic source of carbon, I guess, whether it's zoological or not in nature.
And Nicole, how long is that observing system expected to be in place? Is it in forever, or is it a 3-year study? What is it?
So right now we're funded by the NSF EPSCoR Track 1 program, but we're in year 4 of a 5-year award. And so there's lots of samples that have been taken around the coast and sediments and in the water column at index sites for quite a while. And we are just beginning to figure out how to use and query those data sets. But UMaine has set up a Genomics and Environment Center that's supposed to be continuing in perpetuity beyond the life of the NSF award. And Bigelow's gotten some funding from a private foundation called the Builders Initiative to continue developing some of those probes. But I think the hope is that it's going to be a whole new biological monitoring sort of program that could tie in with or my hope definitely is that it would tie in with some existing carbonate chemistry monitoring programs. But it seems like maybe the time is getting close to bring some of these teams together and see where overlapping sites could be used.
Yeah, I think that'd be great. Maybe we can coordinate a little on that front. Yeah, I'd be very keen on to see where that data is being collected and frequency of observations, who's processing the data, what we can get from the data. Is it any of it aligned in any of the sanctuaries? Is it aligned to any of these, major harvestable resources? I mean, yeah, I'd love to learn more about it.
Okay, cool. Make some connections.
Nicole, if you're not careful, you're going to be roped into being a speaker for one of the upcoming webinars.
I just realized I might have done that by accident. Yes. I think it's a big network of people on that main eDNA program though, so I can find other volunteers that might be better versed than I to talk about it anyways.
But so just to follow up on that though, so right now you're just focusing on macrophytes. What would it take to add in other sources?
Yeah, I am personally, but it's a big group of, I don't know, dozens of PIs, and there are those that are focused on fish and others that are focused on inverts,
Yeah.
and some that are looking at connections between freshwater and marine systems, some that are looking at marine mammals. So we've got a lot of it covered.
Okay.
What's the name of the overall project again?
The main eDNA program. And let me see if I can figure out how to pop the website in the chat for you,
Great.
because I'm trying to do this with my phone.
And Nicole, are they looking just in the water column, or are they looking at the sediment?
Water column, sediment, water column and sediment is it's although we have some people who've done work on collecting DNA from the air as well.
I was kind of jokingly say, what else could there be? But okay, yeah.
Yep. And there are some people who are doing aDNA, which is ancient DNA, so trying to get down in deeper cores too, which is super
Okay,
cool.
that's a lot of DNA. Yeah, we should learn more about this, because yeah, I'd love to know if it can give us insights as to the fate of the carbon that's being generated and where it winds up.
Well, yeah, and there's lots of questions about how long DNA is stable for in each of these environments. So it's not completely foolproof, right? There's issues to be worked out, but there's a lot of promising results.
I noticed we're at the end of time and I really want to get, oh, she's got it. Perfect.
There you go.
I have to go. I don't know if other folks do as well, but I
do
really
too.
appreciate everybody's time like presenting this today. It was just, the talks were really great and the discussion was awesome.
Yeah.
Emily, did you want to close this out?
Sure. Again, thanks for everyone for coming. Thanks, Rob, and thanks, Brendan. Our next webinar is on Thursday, May 4th, also from 1 to 2:30. So check out our website and make sure you're signed up for our mailing list to get more information on those.